Anatomy and physiology are subdisciplines of biology. Anatomy studies the form, the way a body is built, and physiology studies how the body functions, how it works. Anatomy can be studied in a variety of ways. The way we study in this course and the way it's studied in most uh, medical contexts is systemic anatomy. Systemic anatomy means that we are going to focus on each organ system one at a time. We're going to learn the integumentary system, the skeletal system, the muscular system, cardiovascular system, each one of them one at a time. And we are going to go in great depths. We're going to start with the system as a whole, then go down to the level of organs, then down to the tissues that make up those organs, then down to the cells that make up those tissues, then down to the molecules that make up those cells. Uh, and that is the way it's normally done. That's the best way to enter into the field of anatomy if you have no background, because you can learn it in a very systematic way. There are situations in which one would prefer to study anatomy in a different way. There will be topological or regional anatomy. For instance, if you study to be a podiatrist, you're not interested in what's happening in other parts of the body, you're interested in the foot. So you want to know everything about the foot, the bones and joints and tendons and ligaments and muscles and skin and nails and arteries and veins and nerves and lymph vessels, everything that is in the foot. That will be regional anatomy. Uh, that means we don't have to do it here. This is very basic anatomy, so we are going to be studying it in a systemic manner, one system at a time. Anatomy is an observational science. There is not much experimenting in the sense of designing a hypothesis and then testing a hypothesis with an experiment. That doesn't mean we do nothing. We do actually quite a lot. We manipulate the bodies quite a lot. We dissect the bodies, or we take a piece of the body and freeze it and dye it and slice it in order to make a microscope slide so we can look at it in a a great deal of detail. We also use various imaging techniques, X-rays and MRIs and CAT scans and PET scans and ultrasounds, in order to look inside of the bodies, dead or alive, in order to look into the details of the internal organs. So we use quite a lot of manipulation. We use instruments. We use expensive instruments, machines, in order to study anatomy. So it's not passive, but it's an observational science. It's very, very rare that you can come up with a hypothesis in anatomy. You actually dissect and see what you find. On the other hand, physiology is a very experimental science. It is a hypothetical deductive science. What does that mean? That means that you are interested in how does the body do something, and then try to figure out how the organs devoted to doing that manage to do that. And so you're going to come up with an idea. You're going to transform your question into a form of a statement. And that statement is a hypothesis that you're going to try to test. You're going to test it with an experiment. You're going to do, there are all kinds of experiments. Depends, are you working on an organismal level or tissue level or cellular level, molecular level? But let's say that you're working on an organ system level or organismic level. How are you going to set up an experiment? Let's say that your hypothesis is that estrogen is going to be in some way involved with the development of cancer. Let's say that enhances the growth of tumors. And so you want to set up an experiment in such a way that you can figure out if that is true or not. You're going to test the hypothesis in such a way that you design an experiment specifically to disprove your hypotheses. And so, if you fail to disprove your hypothesis, that means that the hypothesis is probably correct. If not, that means that you have to look at some alternative explanations and test them. So, how do you do an experiment? How do you do a controlled experiment that you can trust? Let's say for this experiment, you're going to test it in mice. So let's say you need about 20 mice. You want them to be as genetically close to each other as possible. So siblings out of the same, you know, same pairings of parents, probably from some kind of a mouse colony that has been inbred for many generations, essentially clones. And you're going to take 20 of them, and they're going to be exactly the same age, born on the same day. And then you're going to choose only 20 males or only 20 females. I mean, it's estrogen, it's a sex hormone. You expect it to have different effects on different sexes, so you just choose one sex. And then you take those 20 mice and separate them into groups, 10 in one group, 10 in the other group. Both of those groups, everything you do to them is exactly the same. So you put them in exactly the same cages inside of the same rooms. There's the same temperature of air, there's the same water, there's the same food. The food is changed and the litter is changed exactly the same time of day by the same human. They might like that human, they might not like that human, so that's a stressor. So you want everything, absolutely everything, to be done the same to two groups of mice. And then you want to inject estrogen in one group of mice and not in the other. So you are going to dissolve estrogen. Estrogen is a powder. So you want to dissolve it in order to be able to inject it into mice. And remember that estrogen is a lipid. Lipid there's fat, and fats like oil and water don't mix. That's how lava lamps work, right? Because the water and oil don't mix. So, in order to dissolve uh, estrogen, you have to dissolve it in an oil. Let's say sesame oil. So you're going to pick up each one of those 10 mice in your treatment group, and you're going to inject them with exactly the same dose of the solution of estrogen in sesame oil. Well, you can't leave the other group, the control group, the other 10 mice, you can't just leave them untouched because the treatment group, you have handled them. You have picked each one of them up. You have injected each one of them. And so you have to pick up all of the control mice as well and inject them with something with the same liquid, the same volume of liquid, and the same type of liquid. So it has to be the same vehicle that you used for the estrogen group. So the sesame oil, if you're injecting them with 2 cc of sesame oil, because it's the treatment group, 2 cc of sesame oil estrogen solution. Uh, and So you have controlled for everything you could think of, except one of the groups got estrogen and the other one did not. And then you give them some time, and then see what happens. See if they grow tumors. Give them, I don't know, a month or something, and then open them up and count how many tumors per mice. What is the total mass or diameter of the tumors in each mouse? Um, or See how long they live after the injection. So look at kind of longevity after the injection. So you decide what is the pertinent question, what is the best way to measure, so it actually answers your question. And then you put the data from the control group and the data from the treatment group into a statistical program that will tell you if there's a statistical difference between those two groups in tumor number, tumor size, or length of life after the injection. That can tell you something about it. So most of physiology is done in this kind of way. It can be done with molecules, it can be done with mice, it can be done with humans, it can be done with mathematical models, but you're going to always have some kind of a control and treatment being statistically compared. So it's a manipulative and also experimental, hypothetical, deductive science, unlike anatomy, which is mostly an observational science. The way physiology is usually taught, especially in medical schools and any kind of healthcare directed programs, is systematic or systemic, just like anatomy. We study one system at a time. We study endocrine system separate from immune system, separate from respiratory system, separate from urinary system. And that's how we're going to do this. For a lot of you, this is the first ever encounter with anatomy and physiology, and so we can't go too crazy. We have to go from the beginning, and the best, kind of easiest way to get into physiology is to study one system at a time and learn the details about each system, how the heart works, how the kidney works, how the lungs work. Uh, there are pros and cons to this. On one hand, it's easy, it's systematic. Uh, on the other hand, if you separate those systems, you can lose the sight of the forest for the trees. It's easy to not see how it all works together. The systems, the way we divide the systems, is an educational divide. This is how it was designed in medical schools in probably Germany, 19th century or earlier, in order to make it easier to teach and easier to learn. Those divisions between systems are kind of arbitrary. Uh, we decide if this is going to be part of this system or this is going to be part of that system, because pretty much every organ, pretty much every cell in the body is part of multiple systems. And all the systems work together. No system can work in isolation, no organ can work in isolation, no cell can work in isolation. All the parts of the body are constantly communicating with each other and coordinating with each other, all of their activities. They're influencing each other. And so when you're studying physiology in a systematic manner, system by system, it is a good idea sometimes to kind of step back and to look at the ways the different systems work together, how they interact with each other, how they affect each other. And so we are going to have kind of running themes. We are going to have examples that we're going to repeat over and over again when we go for from one unit to the next to the next. We're constantly going to go back to the principle of homeostasis, see how it works in every system. We are going to look at examples of negative and positive feedback loops in each system. We are going to look for um, countercurrent exchangers in at least three of the systems: cardiovascular, respiratory, and urinary. We are going to uh, barely mention, but we're going to mention scaling laws as if they affect the bones and lungs. We are going to talk about the circadian system and how it coordinates everything. Without a circadian system, all these systems cannot properly communicate with each other. If an endocrine gland secretes a hormone at a time when its target doesn't have the receptor for that hormone, there's not going to be an effect. Those two systems have to coordinate stuff in time, and circadian system is the one that is going to coordinate the timing. That's the conductor of the orchestra. The body is the orchestra, and the circadian clock is the conductor that makes sure that everything happens at exactly the right time. Um, and we are going to talk about the form and function of molecules in a couple of different units. So we are going to try to find ways to show how different systems are working together. One of the kind of running themes that we're going to have is the stress response, acute response, and to some extent, chronic stress response, because every cell in the body is affected by stress. Either it is part of the stress response or is affected by the stress. And once you repeat it for unit by unit by unit, you will never forget for the rest of your life how, for instance, adrenaline or sympathetic system affects any organ. You will always remember that. So, anatomy and physiology are fun, and anatomy and physiology are different from each other, which is why we focus on anatomy in the lab, where you can touch and where you can see with your own hands and with your own eyes, where you can look at your own microscope slides, because it's an observational and it's not just memorizing. On the other hand, physiology is very much conceptual. You need a lot of my talking, my lectures in order to understand it. Reading my stuff, there's going to be additional links occasionally, and there's a textbook with wonderful animations and images to understand the physiology. It's much more understanding, much less memorizing. Uh, there is going to be in Sakai, you'll look into Sakai, there is a subfolder in the lecture materials folder called additional readings. It's a Word file. It's a Word file that you don't have to open. That's fine. There's not going to be a single question on any exam that is referring to that material. But I think you're going to find it fascinating if you go to that file, open it, and every day open one or two links and skim it or read it if you're interested. And I think you're going to get a better understanding of anatomy and physiology and biology and science. You're going to get a better understanding of how different systems are connected to each other, because pretty much every one of those articles is combining two or three organ systems. It's not just about one, which is why it's difficult to organize that file, because pretty much every link, every article or video that is linked there is covering two or three different systems. You're going to also get a feel for how basic this course is and how much more detail there is, which is very, very difficult for any one person to know it all. That's why people specialize. I'm not an immunologist. I know the basics of immunology, and there's so much more about it. And that's about every other part of anatomy and physiology, that there's so much more. You're also going to get a feel for how much it changes. Just because it's in the textbook doesn't mean, it's always been that way and it always will be that way. Things change. There are new studies. There's going to be a sentence or a paragraph in the textbook that is going to turn out to be wrong in the future. And 10 years from now, you look back at it in the textbook and say, hey, this actually, I read about this. This was proven wrong. And we now know better, because that's how science works. It fixes its past errors. Uh, One thing that file additional readings has is links to just Wikipedia pages of people who discovered various structures or processes. Your textbook tends to kind of clean up the history out of it. That's I think that's wrong. I think that we need to learn our science in the context of history. Yes, I know it's mostly kind of white European men. That's how it was in the 16th, 17th, 18th, 19th, even 20th century. So it's not great diversity, but still, those people are interesting. Those people have done a lot of things. That kind of specialization is relatively recent. A lot of them were doing all sorts of different sciences, and philosophy and art. Um, they were interesting characters, and it's very interesting figuring out how they did it, why they even thought about that question. How did they find out that this is how this part of the body works? Or how did they find its structure? What were they actually looking for? Why were they looking for it in the middle of 18th century? So I think it humanizes anatomy and physiology, but also gives you that feel of how it's always changing. And how is it changing? In kind of what ways does the science change? How do we know better than our predecessors? So I hope that you look at those links every now and then and kind of think broader and learn more than just the minimum that your textbook provides. All right, thank you.